Hi, and welcome back to my backyard studio. Today I want to talk about a subject that, in my wildest dreams through most of my career, I never thought that we'd be in a position to speak with such clarity and definitiveness about a subject. And then the subject is the question of how are the orders of insects or hexapods interrelated? This was a pipe dream for many people for decades, for literally centuries. But I think now we're at a point where we have sufficient data and the analyses are sufficiently robust that we can actually think that we have a clear vision of how the major groups of insects evolved. So that's what I want to introduce today. Well, actually, let's start by stepping back a little bit in history and talk about two of the people that I consider two of the monumental figures in insect evolutionary studies over the past century. The first of these is Robert Evans Snodgrass, R.E. Snodgrass. Snodgrass is extremely famous in the field, particularly of insect morphology. He published a classic book called The Principles of Insect Morphology in 1935 that, 40 years later was still the textbook that my professor had us buy and read. Oh, it's painful, but read in insect morphology class. And then some 10, maybe it was 20 years later, it was republished by Cornell University Press by a member of my graduate advisory committee, George Eichwart, and so it's still out there, still available. Snodgrass, not only was he very insightful on seeing homologies in structure among different arthropod groups, even though the details could be so different, but Snodgrass was also renowned as an artist. He had an eye to see the essence of the structure of a creature in front of him, looking at it through the microscope, and then to render that in line drawings that conveyed that essence in a simple, direct, easily understandable manner. I think you'll find that there are lots of subsequent uses of illustrations made by Snodgrass that are still widely in use today. One of the stories about Snodgrass was back in the old days when he would teach a class. I mean, old days when you still had chalkboards, right? That Snodgrass was able to stand in front of a chalkboard with pieces of chalk in both hands and draw with both of them simultaneously to produce really a piece of art on the chalkboard in order to be able to lecture to the students and have them understand what he was talking about. As I said, Snodgrass's primary contribution was in morphology, but in order to understand the correspondences in morphology between the various arthropod groups, inevitably this entailed developing and corroborating or testing hypotheses of the evolutionary relationships among the groups, so they could look at this structure on a decapod and say, well, that's the same thing that we see over here in a dipteran, or things like that. I can't begin to cover all of the contributions that he made, but I want to focus in on one aspect of that, and that is that Snodgrass was of the school, and one of the leading lights in the school, who thought that the arthropods, so everything, crustacea, chelicerates, trilobites, hexapods, myriapods, who am I forgetting? Probably somebody there, that they all form a monophyletic group with all the characteristics that we know about arthropods, exoskeleton, dorsal blood vessel, all of this kind of stuff. And he divided them into two groups, one of which was called the mandibulata. And this was basically those arthropods that have mandibles. Now, of course, not all arthropods have mandibles per se. Chelicerates don't have mandibles. And so he grouped together those that did have mandibles, specifically the crustacea, what we'll call the hexapods for the moment, and I'll get into more about why that's a problematic term in some ways, and the myriapods, so pauropods, symphyla, diplopods, and chylopods, the centipedes. So these guys were all united by the possession of mandibles as opposed to the possession of chelicerae. So he's kind of an avatar here of what for probably from the late 1800s up until the 1970s or so was the predominant theory about how arthropod groups were interrelated, from which insects evolved. That view was challenged by the next highlight I want to introduce to you, and that is Sidney Manton. Sidney Manton was a British researcher. She began her work in embryology, and she made her real claim to fame in studies on comparative functional morphology of arthropods, and specifically how their mandibles work functionally and how they walk, how their bodies are constructed so as to enable them to walk. I meant to mention also that Manton's work is really characterized by just an almost unbelievable level of detail in which she studied the morphology of these character systems, the mandibles and the locomotory mechanisms in particular. And like Snodgrass, she too was an excellent artist. Her artwork did not have the simplicity that characterized most of Snodgrass's work, but it's kind of like going in a different way, trying to convey something different, rather than to try to distill the essence of a system and convey that to the student or researcher, reader, as Snodgrass did. Manton's work emphasized the fine details and the importance of these fine details in understanding how the structures that we see actually function in the world. So the first big work that Manton published addressed this question of the mandibles. Remember, as I just said, the possession of mandibles was one of the characteristics by which Snodgrass united some of the major groups of arthropods, the crustacea, the myriapods, and the hexapods together in one group, and saw them as, although he didn't use that term, he saw them as a monophyletic group. Well, Manton looked at the function of these mandibles, how they worked, and proposed that there were actually two fundamentally different types of mandibles that we find in arthropods. And because she could not envision a way in which some intermediate between these two modern forms of mandibles could have been functional itself, she concluded that meant that there could not have been a single common ancestor of the groups that have these two types of mandibles, because the mandibles that would have had to have would be non-functional. So the two types of mandibles we're talking about here is one that is found in the crustacea and the hexapods, and these mandibles are formed from the basal segment of the limb that corresponds to the mandibular segment of the head. So you remember, one of the characteristics of arthropods is having repeated ventrolateral appendages on each metameric segment, and in the case of the mandibular segment, these appendages have become modified to form the chewing mouthparts that we now call mandibles. Well, in the crustacea and the insects, Manton, by looking at the musculature and the way these mandibles moved, discovered that the mandibles were being moved by the same basic musculature that is used to move the legs on the thorax, or even farther back in the case of a crustacean. That is, there were muscles that caused promotion. I don't have enough room here to illustrate to you, but promotion of the appendage. Promotion, if it's a leg, means to swing it forward so that the leg steps forward. So there are muscles that are pulling on the base of the leg to pull it forward, and then there are muscles pulling on the other side of the articulation to pull it backward, to remote it. So for her, the mandibles of crustacea and hexapods function by this basic promotor-remotor movement around the articulations of the head with the body wall. And by shifting those points of articulation, we could then evolve transverse biting. Remember, transverse biting is biting across the longitudinal axis of the body. So this way. The mandibles are normally moving, where's my hand? In this kind of manner, but by shifting the points of articulation, we could get this such that now we're biting like this, where my thumbs are the biting parts of the mandibles. She called these rolling jaws, with the idea that the inner surfaces, the mesial surfaces where the chewing is taking place, they come together by means of this rolling motion that is fundamentally the same musculature that's involved with the promotion and remotion of the ambulatory legs and the rest of the body. Now contrast that with what she called direct transverse biting, which is found in the myriapods. The myriapods have mandibles, and interestingly, the mandibles of myriapods are segmented. So whereas the mandibles of crustacean insects seem to be formed from the basal segment, because myriapodan mandibles are segmented, that means that they are composed of more than that basal segment. There are multiple segments involved, and she hypothesized that these were constructed from the entire ancestral limb, and so she called these whole limb mandibles as opposed to gnathobasic mandibles. And there's musculature that you can see, for example, if you look at the musculature of horseshoe crabs that moves its leg and their inner lobes of their bodies. I feel like I'm delving deeply into insect morphology, and that's not really the goal today, but I think it's important. There are muscles that connect across the midline, the two mandibles on either side, and by contraction of those muscles, those mandibles are brought closer together, effecting what she called direct transverse biting. So instead of shifting the whole orientation of the mandibles so that now you get transverse biting, these guys evolved by direct musculature there. And so her conclusion was that, well, on the basis of the fact that she could not, what's a good word? Hypothesize, conjecture, imagine a functional intermediate between these two different types of mandibles. That means that they must have evolved these things that we call mandibles today, evolved them independently. And thus, if we were to base a taxon on the joint possession of mandibles, then we would be creating a polyphyletic taxon, because the mandibles that we're basing this group on evolved independently. These are convergent characters and not truly homologous characters. But wait, there's more. Manton also spent, her paper on mandibles was a single large paper, but then she published a whole series, if I remember right, something like 11 papers on what she called the locomotory mechanisms of arthropods and, well, as you can see from this image, related organisms. And so basically we're talking about how the legs function. How do they move? What's the musculature involved with that? How do the legs articulate with the body? And she put a lot of attention, she put a lot of effort into understanding how the legs of this group, the Onychophora, work. Maybe, not sure if you're familiar with Onychophora, or velvet worms is one of their common names. These are a tropical group in the forest floor, in the litter, moist areas. Like arthropods, they have chitin in their exoskeleton, but unlike arthropods, the only thing that is what we might call sclerotized, hardened parts of the body of an onychophorid are the tiny little claws. Let's see, where's my cursor here? Let's make it a laser pointer. Tiny claws at the tip of these. Well, I'll call them legs for the moment, but I'm going to shift to calling these things lobopods, which by their very name gives you an idea of what their function is, that they're kind of lobe-like extrusions of the body that are functioning as legs. So we have sclerotized claws at the tips of the lobopods, and also we have a pair of sclerotized mandibles within the head capsule. And interestingly, these mandibles do not bite transversely. They come out and they scrape back and forth like this, perhaps similar to cycloraphan larvae, fly larvae, maggots, and the way that their mandibles now work today. So, as I said, Manton spent a lot of time looking at this, and basically an onychophoran lobopod works by more or less accordion-like expansion and contraction of these lobopods in order to make them longer. And then there are muscles at the base. See, this lobopod here has been promoted, and this one over here has been remoted. And so not only do we have to be concerned about the kind of movement that we see on the legs, but also when you have this many legs, this has to be able to be coordinated as well, so you're not literally tripping over your own feet. Manton looked at the function of legs in crustacea, in chylopods, in millipedes, in several groups of hexapods. And one of the first things I want to mention about her overall conclusions were that the structures that we see as legs in modern day arthropods actually fall into two different groups. Some of these are just a simple leg that is a simple protrusion from the body, kind of like the lobopods on this creature, except you have to incorporate sclerites and the joints between the sclerites. This is contrasted with the legs that you see, for example, in the crustacea, where each leg is actually two branches, an inner branch and then an outer branch, and sometimes there's multiple outer branches on each of these segments. And that distinction, she coined the term for those that are composed of just this simple single branch. She called that the uniramia, and the biramia were those that had an inner walking leg, and then typically this second branch of the leg would function as a gill or something along those sorts. So again, she concluded that what we are seeing in modern day arthropods, and when we look at their legs, are actually legs that have evolved independently from one another, that they did not evolve from an ancestral sclerotized segmented leg, again because she could not conceive of any functional intermediates, but they each evolved independently from a lobopod-like appendage, not specifically an onychophora. Onychophora, she thought, was one of these very old groups that retained that ancestral character state. But that the two types of legs, the uniramous leg or the biramus leg, along with the sclerotization and musculature patterns within them, evolved independently within arthropods. Another way of saying this is that arthropods, the common ancestor of what we see as arthropods today, was not itself an arthropod, and therefore the concept of the taxon Arthropoda is itself a polyphyletic taxon. She thought arthropods were polyphyletic. And this, by a combination of things, by a combination of the exquisite detail and insight she had into these organ systems, her artistry, her ability to convey that information through her artwork, and the fact that she was I say this kindly, very adept in the art of polemics, in arguing her case, in giving the exquisite put down of her detractors. She was an excellent arguer. Because of a combination of all things, Manton's ideas gained prominence for a whole generation of entomology and arthropodology, such that it was commonly accepted for 20-25 years that what we today consider to be just a bloody obvious monophyletic group, the arthropods, in fact that was a polyphyletic group. She also, among the hexapods, she looked at the function of the legs in the microcoryphia, in the Thysanura, in the locust, the grasshopper-style locust, and also in Collembola. And interestingly, she found that the way that these different groups of organisms move their legs suggested to her, recurring pattern here, that there could be no intermediate state between the endpoints that we see today. That itself could have been functional, and therefore the legs that we see in the Protura, in these groups that I mentioned, must have evolved independently, and therefore that the arthropods that have 6 legs, the so-called hexapoda, are themselves a polyphyletic group. That Collembola evolved their 6 legs independently of the true insects, the Thysanura plus the Pterygoda, the winged insects. And so sometimes, very commonly actually, you'll see people distinguish between the concept of hexapods on the one hand and insects on the other hand, where hexapods may be polyphyletic, may be monophyletic, and we'll get into that in just a second. That's one grouping that includes all the winged insects, the Thysanura, then the microcoryphia, and the 3 orders of Entognathous insects, the Protura, the Collembola, and the Diptera. Those are the hexapods. And insects, the true insects, are restricted to the Thysanura plus the winged insects. Now, one of the weak points in her argument was the assertion that the ancestral character state, the character state possessed by the ancestor of 2 different insects, must have been an intermediate in some form with what we see today. And that's not generally accepted any longer. It was accepted for a long time by virtue of the force of her arguments and the body of evidence that she marshaled for her argument. But that really is not an effective argument. If you're going to say that a group is polyphyletic, the telling argument is not to say I don't see how we could have had an ancestor that combined the characteristics of these 2 groups, but rather to say, well, this piece of the group is actually more closely related to that other set of species over there, to show that they have closer relatives elsewhere outside the group. And so once that mode of thinking came on, I mean, her work was done contemporaneously with Hennig and the rise of phylogenetics, but these things never really met in her work. Matter of fact, maybe I remember some of her writings where she didn't buy into the Hennigian system, at least in some aspects of it. But anyway, these did not overlap. And as the mode of thinking associated with phylogenetics took over, we saw that it came to be interpreted that Manton's work was flawed in that way. Not the underlying data, the underlying interpretations of how these legs work, how they were put together, and how they moved, but what that meant for the evolutionary patterns within arthropods and in insects in general. So basically, Manton is a proponent of the idea of polyphyly in its many forms. Arthropods are polyphyletic, hexapods are polyphyletic. whereas Snodgrass and his ilk thought that basically we had two groups, the chelicerates and the mandibulates, that were the basic breakdown of arthropods. So let's move on to the present day. Oh no, not the present day yet. I just wanted to show you this illustration, which is amazing in many ways for how similar this, well, let's call it a phylogenetic tree for now, how similar this tree is. Published in 1904 by a German researcher named Berner. How similar it is to many of the modern ideas we have about arthropod relationships. Let's see, let me kind of guide you, walk you through this tree here. So we have 1904, so this is pre-Hennig. This is pre-molecular data. This is based totally upon fossils and external morphology and interpretations of those things, and trying to see how they the data can fit together into a coherent hypothesis. So Berner has an ancestor that is a crustacean, which we can argue with or not. He has myriapods coming off as two separate branches here, with the centipedes over on the right here, and the diplopods, the pauropods, and the symphylans over on the left. Then we get into the hexapods per se, and as the representative of the ancestor of all hexapods, he's got the name Protentomon. That is a name associated with the modern hexapod order Protura, and from which we'll talk about proturans next time. But proturans are unique among all hexapods in the fact that they have no antennae. They also have no eyes, but that's not unique. But the combination of no antennae, no eyes, and some other developmental characteristics makes it easy to interpret in some ways. These are creatures that are retaining very ancestral features of the arthropod body organization. Moving on, just quickly, we don't need to get into too much detail here. On this branch on the left, we have the remaining Entognathus orders. We have the Columbella or springtails up here, and the Diplura down here. On the other side of the tree, we have the remaining two orders of primitively apterous insects that you saw in the lab. We have the Zygentoma or Thysanora, it's the same thing here, and the Archaeognatha down here. So these are the jumping bristletails and their relatives here. And then from this branch here, wrote it upside down for some reason, but this is the pterygotes. These are the insects with wings. So remember, this is the stuff that originated sometime between the Devonian and the Upper Carboniferous era, we think. Getting into the tree here, I don't want to get into all the details, but we see a massive grouping over here that includes the true bugs, and the bark, book, and true lice over here. You'll often see this clustering of orders called the Paraneoptera. So I guess I jumped the gun here a little bit, since Neoptera reminds me, this branch here leading to the Odonata and the Ephemerata here, this corresponds to the Paleoptera. So Paleoptera going off to the right here, and the short branch here that comes up immediately represents the origin of the Neoptera, the ones that have the third axillary sclerite that gives them the capability of flexing their wings. Notice that Berner has the Paleoptera as a monophyletic group. Within the Neoptera, we have, as I got ahead of myself a little bit there, the Paraneoptera over here. And then over here, nominally as a monophyletic group, although the connection of the branches here gets a little bit tight and it's hard to represent. Here's what you had in your last lab, the Polyneoptera. So we have walking sticks, we have the Orthoptera, the jumping Orthoptera, we have Oophthecaria, I think that says, the mantids and the cockroaches, stoneflies or Plecoptera, earwigs, Dermaptera. Who else do we have here? It's a little bit, Isoptera. Interestingly, not placed particularly close to the cockroaches, where we think they are today, and then the web spinners, or Embioptera or Embiodina here. So this is the Polyneoptera, hypothesized by Berner, although he didn't use these words, as a monophyletic group. And then this last long branch leads to the holometabolous insects. And we're not going to worry about the details about relationships in there. We'll get to that a little bit later in the course. This has a number of remarkable similarities to some of the ideas that prior to the paper we're going to discuss and that you read for today were common hypotheses out there, and people were trying to find evidence to decide amongst the various hypotheses. But this was already articulated in 1904. So now let's move forward to the present day. And as I said, that it just boggles my mind that we're in a position to actually have on the front page, on the cover of Science, the subtitle Insect Phylogeny Resolved, Answered is the implication here. I'm always going to put a little bit of hesitancy in saying, yeah, we know, but we certainly have a major amount of evidence now to talk about how all of these insects are interrelated. And so we're going to talk about insect phylogeny and the One Kite project. So, what is the One Kite project? Basically, it's a project to try to infer the relationships amongst species of insects on the basis of molecular data, and specifically on the basis of transcriptomes. So just as a very quick review of transcriptomes, basically in order to do one, you extract RNA from your material, reverse transcribe that into cDNA or complementary DNA, and then you sequence this complementary DNA. In your analysis, I mean, you're going to be sequencing all sorts of RNAs in there, but the analyses typically focus on the transcripts that have these poly A tails, indicating that these are derived from messenger RNA. So what you're sequencing are the protein coding genes that are being expressed at the time that the RNA was originally extracted. Let's see, protein coding genes. So they have to be being expressed at that time. So you're not getting the full suite of things that are encoded within the genome. You're also not getting, probably not getting exons. Let me see if I can get this right. No, you're not getting the introns. What you're getting are the exons. I always have to think about that. Because those are probably already spliced out. And typically each transcript that it's sequenced, you're getting a single, this is a transcript of a single gene. This is not true in the case of some mitochondrial genes where the mRNA can actually be multiple pieces of different genes, which then somehow later get cut apart and go to their merry way where they belong. But typically it's the transcript from a single gene, and one also has to be careful of the issue of having alternative splice sites within the gene. In other words, that an organism might have one gene with 7 exons, but can mix and match those 7 exons in different ways in order to make a functional protein at the end. So it's possible to have different transcripts coming from the same gene, all making for, methodological difficulties, but fundamentally that's the data that we're getting. So once you have your sequences, then we go back to the same route that you're now very familiar with. Align, identify the homologs amongst the organisms you're studying, align the sequences, partition them if need be. So different genes or different parts of genes may want to say evolve following different evolutionary models of DNA evolution. And so you're going to break them into chunks where we're going to apply one model to this chunk and another model to that chunk, and then do your analysis. Your analysis can be, all the usual suspects: maximum parsimony, maximum likelihood, Bayesian techniques, or coalescent techniques. All of these are fair game. So that's what the data is coming from. In the One Kite project, now One Kite itself is an acronym, a little bit strained acronym, but it's an acronym that stands for 1000, 1K, insect transcriptome evolution. So basically the idea was to generate 1,000 insect transcriptomes and then analyze them. It's still active. The One Kite group. This is an international collaboration with the lead organizations being a couple of in Germany, and you can probably, even if you have never seen a word of German in your life, figure out what these things stand for, except for the word Forschung. Forschung means research. So the Zoological Research Museum of Alexander Koenig. Koenig means king, so King Alexander or Kaiser Alexander. And this is the Center for Molecular Biodiversity Research. So those are in Germany. And then the BGI is a massive sequencing operation located in Shenzhen, China. So Shenzhen is just outside of Hong Kong in the province of Guangdong. So collection of organisms for extraction, reverse transcription. The cDNA is sent off to BGI, where they have, thousands, literally thousands of Illumina sequencers going nonstop, and the technicians to run and babysit them all the time. The data comes back, and their analyses, or basically maximum likelihood analyses, and the software engineers for RAXML and XML are also closely associated with this project. I didn't check, but I believe if you look at the list of authors in the paper you read, you'll find Alex Stamatakis somewhere in the long list of co-authors on the papers. So they all collaborated on the project to look at overall insect evolution, so the relationships amongst the orders of insects. And then there are numerous sub-projects focusing on a single order or cluster of orders, in the case where you have things like Polyneoptera, where you have lots of orders. Personally, I'm not directly involved with any of the groups, or the Hymenoptera group or the major group, but I am familiar with some of the participants in that. And because their data are open access, we're able to scarf up some of the transcriptome data that went into these papers and use them for our own studies. So that's what One Kite is. A little bit more about the data and analyses that went into that. So for this study that you read, this represents the transcriptomes of 103 species, representing all orders of insects. Now, if you think about taxon sampling issues, how well is the diversity of all insects being represented? 103 species out of 80 million, or whatever number you care to name. That's pretty modest, but if judiciously done, and in this case I think it has been judiciously done, then the results can be robustly interpreted. And when I mean judiciously done, one of the things to be sure of is that insofar as possible, and as you'll see it, or maybe you did notice that it wasn't always possible, but to have multiple representatives of the major lineages within each order, not just have a single one. They had chelicerates, a small group of chelicerates, myriapods, and crustaceans to serve as their outgroups. And the data that they ended up with, it's presented in this paper, is sequence data from 1,478 single copy nuclear genes that are present in all 103 species. So if you think about it, we're talking about trying to decipher the relationships amongst orders of insects that diverged at least something on the order of 400 million years ago. So in order to find genes that all of these guys share, we're talking about very conservative and slowly evolving genes. If they were evolving too fast, you wouldn't even recognize them as being orthologs. the fact that you can get nearly 1,500 of these things that are found across the entire range of the organisms you're studying gives you a lot of power in interpreting the results. 1,500 genes corresponds to about 413,000 amino acid sites, which if you then simply look at how many nucleotides you're talking about, we're talking about on roughly 1.25 million nucleotide sites for analysis. So a massive amount of data. They analyzed their data. Partly it was philosophical, and partly it was practical, but their analyses are based on maximum likelihood inference. So as I said, RAXML or XML. And they did it a couple of different ways. They analyzed the DNA sequences, but concentrated their attention on the second codon position in these protein coding genes. Remember, it's possible to have synonymous substitutions of nucleotides in the first and particularly in the third codon position. And just to reiterate what I mean by a synonymous substitution is that in a codon codes for a particular amino acid. If you change a nucleotide at a position and that still codes for the same amino acid, that would be a synonymous substitution. So there are lots of synonymous substitutions in the third codon position, and there are some, definitely a smaller number, but some in the first codon position. But any change in the second codon position results in a different amino acid being translated from that. So that's one method of analysis. Another method of analysis was simply to go in directly and look at the amino acid sequence. Looking at the amino acid sequence gives you more character states. Rather than just 4 nucleotides for a position, you have 20 amino acids that things can be. There are still some functional constraints, hydrophobic versus hydrophilic amino acids and the like, that are involved in that. And there's of course different models of how amino acids evolve as compared to how nucleic acids evolve. And then one of the noteworthy things they did with these data, they didn't just run the analyses and say, well, here's my results. They went back and painstakingly looked for what the impact on their results might have been from the violation of some of the underlying assumptions that are going into their analyses, things like uniformity of substitution rates and things like that. So that's the background. Let's dive in and see what they found. I have to say the magic words. I know you can't read this, but you have a PDF where you can zoom in as much as you want. But basically, here's their cladogram showing the relationships amongst insect orders. I am going to zoom in and focus on some things here in the next few slides. But some things to point out here, we have that it's dated here, and this is expanded a little bit here for easier interpretation, showing when their molecular data shows that the different insect groups, insect orders evolved. Remember, this is typically, molecular dates of origin are typically older, sometimes significantly older than dates that are based upon the fossil record. Part of that is simply due to the nature of the fossil record. The first appearance of an organism, it's probably not going to be fossilized. It will be some later generation that gets fossilized. But also some methodological things also come into play here, that molecular data give you older dates. The support values for the nodes are color-coded. You can't read it here, but when we focus in, I'll be able to tell you a little bit more about that. What else do I want to say? The groups are aligned here with a rather artistically inspired representation of the organisms, and with the outgroups at the top. So we're going from the base of the tree here toward the apex, toward the top. So since we're talking about the major relationships among insect orders, let's take a look at some of their results. I'm not going to get into the fine details. We'll do that when we talk about the individual orders starting in the next video. So Here I've zoomed in on the base of the tree, and in this bright sunlight I can't quite see the colors, and I'm partially colorblind anyway, but I believe that's a red dot above my red laser pointer dot, and that is the highest level of bootstrap support for nodes. So I believe that was above 98% or above. So these are bootstraps, so that's a really, really high value as compared to a posterior probability. So let's see, here's this line here. Anything above this are outgroups, and below this are the hexapods. And so the first conclusion I want to highlight here is that, contrary to Sidney Manton and the idea that reigns supreme in entomology and in zoology from the 1970s probably up till the 1990s or even later, they conclude that hexapods are indeed monophyletic, and that's this node here, node 130. So on this side of the node we see Aceryntomon, that's a proturan. We have some Collembola, then we have the Diplura down here. Hexapods are indeed monophyletic. I suppose you can still argue that they evolved the hexapodous condition, that is, using only the 3 pairs of legs as their primary locomotory organs independently, but that is a bit of a stretch. It's a simpler hypothesis to go with, that because they're a monophyletic group, and because they all basically rely on 3 pairs of legs, then that is a plesiomorphic or an apomorphic character for the entire group, defining this as a monophyletic group. Okay, so that's the red arrow. Hexapods are monophyletic. We can lay that baby to rest. The second here, so in lab you saw some representatives of the so-called Entognathus orders, and I'll have lots of good pictures for these in the next video. These are basically 3 primitively apterous groups of hexapods: the Protura, the Collembola, and the Diplura. And the question is, how are these 3, or the question has been, how are these 3 groups interrelated, and how are they related to the rest of insects? As in most such cases, every possible pattern of relationship has been proposed in the literature and has had evidence identified to support each one of those hypotheses. And so we had really been in a quandary as, is this evidence more important than, more meaningful than this evidence? On the other hand, how are these things interrelated? And it impinges on ideas about things like, well, when did the compound eyes of insects evolve? And things like this. When did the ectognathus, the external mandibles, evolve? Did entognathy evolve more than once? Well, to come to the rescue of our older confusion, the One Kite project, with the greatest, with over 98% bootstrap support, comes down on the side of the hypothesis that Protura and Collembola are each other's closest relatives. This combination of orders you'll see in the literature as the Elyplura. I'm not going to expect you to remember the name Elyplura, but if you do see it, should be able to harken back to, oh, this is one of the hypotheses about how the Entognathus insects are interrelated. So Protura and Collembola are each other's closest relatives. Note, however, that Protura are represented only by a single genus here. I don't know how. They got enough RNA, or maybe they had a colony of Proturans. I don't know what they did to get enough RNA in order to do a transcriptome of Aceryntomon. That is a technologically amazing feat to me. So that's the second conclusion. Protura and Collembola are each other's closest relatives, leaving the Diplura on the outside. And here we see Campodia. Can't quite read that. Japix is the root of a number of genera in the Diplura, leaving the Diplura here as the sister group to the rest of insects. Third conclusion. This might seem like a minor one to you, but it does address a rather prominent assertion that was in the literature. They found that the Thysanura are monophyletic group. Thysanura also goes by the name Zygentoma. And the issue was this baby here, Tricholepidion. Tricholepidion, by one of the prominent insect morphologists, E.L. Smith, was thought to be actually more closely related to the winged insects than they were to other members of the order Thysanura. The One Kite project, however, again, 98% bootstrap support, comes down with on the side of the hypothesis that the Thysanura are really a monophyletic group. There's the Zygentoma there. Next, we talked about this last time, and when we were talking about fossil insects, the idea of Paleoptera. Are they monophyletic? Are they not? Here we see that the One Kite analysis proposes that the Paleoptera are monophyletic. So here we have Calopteryx, one of the damselflies, and we get Chordalogaster, Calopteryx as representatives of Odonata, and then down here we get the Ephemeroptera, the mayflies. It comes to the hypothesis that it's a monophyletic group, but you notice there's no colored dot here. That means that the bootstrap support for this hypothesis, in fact, was, let's say, relatively low. Lower than, I mean, most of the dots here, there are a few that are not red, but most of these are red, giving the highest level of support. And certainly when we go farther down on the tree, they're almost all red. But we don't have the same strength of support in the data for the hypothesis that Paleoptera is a monophyletic group. So while they present that conclusion, it seems that even with 1,478 protein coding genes, 1.25 million nucleotide positions, that's still not enough data, and perhaps there never will be enough data to definitively conclude one way or the other concerning the relationships of Odonata, Ephemeroptera, and what we know as the Neoptera. One possible rescue for this would be to increase the number of representatives within each of these orders. That may help to resolve some ambiguity in the sequence data and come more strongly down on one side of the issue or another. I'm sure that's one of the foci of one of the sub-projects of the One Kite project that is still ongoing. So let's see what we have next. Well, if we got Paleoptera on one side showing up as a monophyletic group, not surprisingly, everybody else with maximal bootstrap support here, everybody else comes out together as a single monophyletic group, which goes by the name of Neoptera. These are the ones that flex their wings by means of rotation of the third axillary scleroid of the body. An old idea originally based upon morphology, but now finding corroborating support from this new extensive molecular data set. Surprisingly to me, the Polyneoptera come out to be a monophyletic group. This was probably mentioned in the lab. I know of no morphological characteristic by which you could identify an insect as a Polyneoptera, and that is no morphological feature, no feature of structure of its body that you could say that's a Polyneoptera and it's not anything else, and that you could say that about anything. Yet the molecular data with maximal bootstrap support here confirm that hypothesis. or another way of thinking about it, is rejecting alternative hypotheses. It's saying the maximum likelihood estimate of the phylogeny here has the Polyneoptera as a monophyletic group, which is very interesting to me because there's a lot of disagreement, not only that this is monophyletic, but what the pattern of relationships is in this mess of orders that we have here, because there's a whole bunch of them. What do we have? Zoroptera, Dermaptera, Plecoptera, Orthoptera, Manophasmatodia, Gorilloblattodia, Embioptera, I call them Embiodyna, Phasmatodia, Mantodia, Blattodia, Isoptera. As we'll get to later, you'll see that Isoptera fall within Blattodia, so this is really one monophyletic group. And then with Thysanoptera, we're into the next branching down here. But to me, a very surprising result. Polyneoptera is retrieved as a monophyletic group in this analysis. Remember that 1904 phylogenetic tree of Berner that I began with in this video, where they had the lice and the true bugs all coming in, and I don't know if I mentioned it, but also the thrips coming out together as a monophyletic group called the Paraneoptera. Not so in the one-kite analysis, for we see that on this branch here, subtended by the blue arrow, we have the thrips and we have the true bugs. But what they're calling Socodia, which your book has as the Socoptera, plus the Theraptera, the true lice. So we're talking about the bark lice, the book lice, and the true lice, both the chewing and the sucking lice. They're coming out separate from the rest of the insects with piercing, sucking mouth parts. Now, to be sure, the type of mouth parts is not a characteristic that united these things as Paraneoptera, because bark and book lice have chewing mouth parts. Chewing lice obviously have chewing mouth parts. And one could make, I think, a very reasonable argument that the form of the mouth parts in the thrips is different enough from that of the true bugs to have evolved independently there. But the main point is that while many analyses talk about Paraneoptera here, this analysis did not come up with that conclusion. It found that the thrips and the true bugs do in fact form a monophyletic group, but that the bark lice come out separately, again, maximal bootstrap support here, as a monophyletic clade. Which leaves us with everybody else, which are the holometabolous insects, those insects that have complete metamorphosis. Another term that's commonly used for this is the Endopterygoda. So use them. They're used synonymously. You have to be a little bit careful because when you say complete metamorphosis, we're usually talking about, well, we have a larval stage, then we go through a pupal stage and come out as a reproductive adult. Well, in fact, there are intermediate stages between immatures and reproductive adults that look for all the world like pupae and act for all the world like pupae in a couple of different places in this tree, not only appearing here with the holometabola, but up here with the Thysanora, and somewhere in here in the Sternorhynchus true bugs, the scales and things like that. So as one t-shirt from the Cornell Entomology Club has it, it's got a thrips there saying something like complete metamorphosis before it was famous. Or so I'll have to find that t-shirt and wear it sometime for you. So those, I think, are the major take-home messages. The overall take-home message is we now have strong, I consider very strong support for some of these hypotheses that before were based upon highly suggestive but frankly quite minimal amounts of support from data derived from morphology. Now with this massive molecular data set, we're getting corroboration for a lot of classical ideas, which gives us great confidence that they are right. Both morphology and molecules are pointing in the same direction. And also some ideas of how to resolve some long-standing ambiguous points. The relationships of the Entonathus orders are, well, it didn't resolve the Paleoptera issue, but just focused attention on it yet again. And then Paraneoptera, for example, actually refutes a long-standing idea. So let me briefly summarize. Drum roll, trumpet, blare, insect phylogeny is solved. Well, or maybe we're closer. I think it's safe to say we're a heck of a lot closer now than we were 5 years ago, or I guess it's a little bit longer. This is published in 2014, so let's say 10 years ago. The major conclusion from this one kite project at the level of overall phylogeny of insects is that hexapods are monophyletic. Collembola are the closest relative. The closest relatives of Collembola are the Protura, and that's the group called Elypleura. Maybe Paleoptera is monophyletic. The evidence points that way. There's some room for equivocation. Let's say there. So more research is needed there. Neoptera is a monophyletic group. I don't think there was any doubt about that, but here we have verification of it. Interestingly, Polyneoptera comes out as a monophyletic group. On the other hand, Paraneoptera does not, because the lice do not appear to be most closely related to the true bugs and thrips. They come out as most closely related to the final group on my list here, the Holometabula, Aremdopterygos, the insects with complete metamorphosis. So the plan now is to continue on looking more closely at the insect orders. Well, we'll group some of them together because we have some small orders, and there's no point, no way to spend an entire video talking about just one of them. But that's going to be the plan for the remainder of the semester. So enjoy the beautiful weather. I hope the birds weren't too loud and noisy, and I will see you next time, same bat channel. So long.